this is Miss Dagenford, and we're going to draw a Calvin cycle. And I know, I'm hoping the Google Slides are super helpful, but I also know it's very helpful for this concept, especially to actually draw something. Now, if you would rather draw this on a piece of paper, knock yourself out. As you're watching the video, we can always hit pause at times that you need to. But I think it would be helpful to do it on the Jamboard first, and then you can always pull out a piece of paper and practice it yourself. So that might be something to do as we finish. So let's get started with some reminders. The Calvin cycle is the name of the cycle that's in the light independent reaction. That's going to be happening in the chloroplasts in the stroma. So let me switch to slide 3 real quickly just to remind you. Here's our thylakoid membranes. Stunning artwork, I know. Here's our thylakoid membranes in our light dependent reaction that we learned about. And remember, the goal was to make ATP and NADPH. These are two energy carriers. ATP is going to carry it in the phosphates. NADPH is carrying our high energy electrons and a hydrogen. These are hot electrons, if you will. They are bursting with a little bit of energy. And so let's go back to our diagram. And so this is happening in the stroma of the chloroplast. So let's start out. We've got our 5 carbon molecules. Now, these 5 carbon molecules, and if you can write with your pen, that's fine. I can, but you can also do a text box. These 5 carbon molecules are called RUBP. Now, that doesn't really make much sense. It's not like calling it sugar or proteins. RUBP stands for ribulose bisphosphate. It is a type of it's a molecule related to sugar, but it's not sugar for the plant, and that's important. Now, that's fine. We can call it RUBP. That's all we need to worry about. The thing that I want you to notice is that they have 5 carbons. Now, the first thing the plant wants to do in order to make sugar is to get carbon. That's going to add mass to the plants. It's going to add molecules to the plant, and the plant's going to actually be able to grow. Now, the way it's going to do that is to grab carbon dioxide from the air. Now, what can be really annoying for students, and I get it, in order to keep diagrams from getting too cluttered, they simplify, and sometimes they simplify too much. These carbons are actually carbon dioxide. Now, if that helps, that's fine, but you can see if we keep adding stuff to our carbons, it gets real cluttery real fast. So just understand that we're going to be adding CO2, carbon dioxide, to our 5 carbon molecule. So I'm going to draw a nice big arrow over here. I actually wanted a highlighter there. Make it a little thicker. There we go. So I'm going to make a nice big arrow. And what we're simply going to do is grab these carbon dioxides and attach them to our RUBP. So literally what we're doing is we're taking these carbon dioxides and adding them to the RUBP. Now, what we end up getting is a amazing math here, guys. Hardest math question of the day. We're going to get a 6 carbon molecule. We had a 5 carbon molecule. We added carbon dioxide to it, and we're going to get a 6 carbon molecule. I know, tough, right? So that's what the plant does. Now, this doesn't just happen, right? Carbon dioxide in the air doesn't just like spontaneously form these molecules. So we need something that's going to speed this process up. And did we learn about something that speeds up reactions? Why, yes, we did. We have an enzyme that we learned about in the other video that is going to take, and feel free to you can just draw something similar. But we're going to have an enzyme that can take this RUBP and the carbon dioxide and connect them together. Because this does not happen fast. I'll leave that there just so you can see it. So that's what's happening in this first step. So let's go ahead and move the sticky note over. Carbon dioxide attached to RUBP. Rubisco helps. So then we get these 6 carbon molecules. Now we're going to do a little bit of an accounting here because I want you to see what's happening. These 6 carbon molecules break down pretty quickly into 3 carbon molecules. I know, some more tough math. Uh, so, and we can have a little fun and twist them around if we want. But these 3 carbon molecules, they're still not glucose. And so what the plant is going to do we need to add some stuff to it. Remember, we're making glucose. We're going to add some things to it. Well, let's go back a step. It's going to break down, and then we're going to add some things to make this happen. So what are we going to do? We're going to add some ATP. Let me move these over just to give some more space here. We're going to add some ATP and NADPH. Let me just scoot these up just a little bit so we have a little space there. So along comes ATP and along comes NADPH. I'll put that one there. And they're going to dump their energy into this process. NADPH is going to dump its electrons and hydrogen. And remember, those electrons are hot. They're energy. And then ATP is going to provide its energy. And when they do that, notice, and I was trying to be sneaky here, notice now if I add ATP, I will have ADP and a phosphate. That's what happens. Remember, when ATP gives its energy, that's what we get. So part of the question is, what happens to these guys when they're done, when they've added their energy? NADPH dumps its hot electrons, and now we've got this empty dump truck. Think about that for a second. Where could they go to get more hot electrons, and where could the ADP go to get back to ATP? All right, well, we're almost done here, guys. We're adding our hot electrons and ATP, and we're converting these 3 carbon molecules to a slightly different 3 carbon molecule. And I know that's a little frustrating to just say it went from purple to yellow. So one kind of nice thing is the diagram that I used does kind of add a little teaser, like what's happening. So remember, NADPH is going to dump electrons and hydrogens. And so we can turn these carbons that have no hydrogens, and we can add the hydrogens, and remember, we're making glucose. Now, you're not going to have to explain that whole process, but the point is we took some 3 carbon molecules, they're called PGA, phosphoglyceraldehyde, and we turned them into another molecule. And that's what's kind of key. So let's add our sticky note. We're going to add energy with ATP and hot electrons. And now what we're going to be able to do is pull one of these, they're called G3Ps, glyceraldehyde 3-phosphate. Again, you can know it as G3P. Basically, this is almost sugar. We're going to pull some out, and we're going to make glucose. And you might be thinking, smart person that you are, wait a minute, if we need G3Ps, why didn't we use all of them? And that's a good question. What ends up happening is this cycle has to happen twice in order to get two G3Ps that we can use to make glucose. The rest of these G3Ps get recycled back to RUBP. And this can be terribly inefficient looking to students. And I get it because we went through all of this trouble to make all of these G3Ps, and I just told you that that's what we need to make glucose. And then you're telling me that we're not going to use them to make glucose. But here's the trick. This process is so energy demanding that we need as many reactants and many carbon dioxide and many Rubisco in order to make this happen. And so we need a lot of these RUBPs, a lot of the carbon dioxide, and a lot of the Rubisco enzymes. Think about a big boulder that's going up a hill. The more people you have that can add to it, the better. So the plant is designed to take a lot of those G3Ps and convert them back in order to keep the system going. Otherwise, it's just going to stop. So the whole thing is going to happen one more time, and then you're going to get another G3P out, and then you have glucose. Now let's take the last little minute we have and see if we can answer the question of what happens to these guys. There's my dog again. And let's go back to our slide 3. So remember, in our light-dependent reaction, we made NADPH and ATP. They're going to go over to the Calvin cycle and drop off their energy. NADPH is going to drop off their hot electrons and hydrogens. ATP is going to drop off their phosphate. Now they're empty, but guess what? They can go back to the light-dependent reaction and keep going. They can get restored back to ATP and pick up more hot electrons, and it just keeps working and working and working on the order of billions of molecules per second. So hope that helps. I gave you a blank slide so you can kind of go back and make it nice and neat and pretty if you want. I know mine, if you're following along with mine, it may not look as what you want it to look, so you can use that blank one to kind of redo it. So hope that helps with learning the Calvin cycle.